Welcome to Taxonomy Tuesday. This is March 11th, 2025. We are a service of the New Jersey Mycological Association. We get together every week to share our observations of mushrooms and talk about them and discuss other interesting things related to mycology. So far I have on my list of people to share Doug, Tristan, Christopher, Brandon, Lila, and Hunter. Am I missing anyone? If not, why don't we start with Doug? And let me set up sharing here. Okay, you should be able to see my screen now.
Yes, this I saw at the very end of October. Since it's been so dry, I had to dig back to find a something I thought would be interesting. I know one of the people in the club knows quite a bit about slime molds, so I'd like them to maybe comment a little bit. I ID'd this as Reticularia splendens. I came up with three different families that it was classified in three different websites. Lycogaliaceae, Tubiferaceae, and Reticulariaceae. So I'm not sure what's going on there. What you see here is it grows on well-rotted dead wood. And the kind of tough cinnamon brown covering, I believe, is referred to as an athelium, and the white is the remnant of the acellular slime mold Plasmodium. It's sort of sitting on that white base as it was migrating. Now it's had some kind of signal, and it's creating this athelium-like structure. It's kind of interesting. I hadn't really seen it before. So maybe someone could comment about, what family it might be in, and have other people seen it? I suspect yes. And yeah, I mean, it's an acellular slime mold, and that's probably all I know about it. I couldn't find a great deal of information on it other than that.
Our slime mold expert, Virginia, is not yet with us.
Oh, okay. So
I think that's a pretty good idea, though.
yeah, it looked like it, from the looking at the various websites I could see. I'd just like to find out about a little more about, maybe learn a little more about slime mold classification. I know, of course, it is not it's in the protozoa. It doesn't have chitin like Basidios and Ascos, so it's a totally different class. And
Yeah, and I don't know much about the taxonomy of these things, or what family, how the families have been moved around. But I do know, in talking to John Dawson, who recently got into slime molds, he was telling me that it's a recent kind of divergence into new families and different actual kinds of slime molds. So like true slime molds, and then there's three other, there's true myxomycetes, and then there's three other things that we call slime molds that aren't true myxomycetes. And that work has been done recently, so things are probably moving around a lot. But again, that's just from a conversation I had with one person.
yes, I got that impression. It's in flux, and it's a kind of a paraphyletic group, meaning they don't necessarily have the same origins under the common name slime mold. But maybe if it's Virginia, maybe if she'd be willing, she could give us at a future talk, since these things are interesting, just a little more background about them and what we might expect to see during the various seasons that maybe Mike, you could ask her, or someone could ask her in the club.
Sure. I think that's a great idea, Doug.
Is that one dinged up because you poked at it?
No, I didn't poke at it. It is you know, it was starting to dry out. It had rained a little bit at the end of October, and I believe that's probably how it's releasing its spores. Apparently, it has dark brown spores, and
Yeah, you kind of caught it. So this thing has, yeah, and it goes through its different stages. You know, it goes from like its plasmodial stage to its mature stage in a matter of hours.
yeah,
And you caught it kind of right in between, because when these things reach maturity, they're not very slimy. They get a hard exterior shell, and that kind of cracks open and releases powdery spores.
yes,
Yeah.
More
So,
like dog vomity.
yep. Yeah. Kind of like when dog vomit reaches its maturity, you know, it's very dry looking, right?
Cool.
yeah. A lot of them produce these small little knobs, hundreds of them, but this one is contained in this egg-shaped thing called an athelium, a single large structure, and apparently that internal thing is called a pseudocapitulum. So the
Yeah, and
thing,
I like how you can see on the one on the left the kind of snail trail, right? As he kind of
yeah, yeah,
shifted along looking for food.
yeah. I mean, I would guess that the drying out or the lack of food is a signal for spore to make the structure and release your spores. But I'm not sure, all the various signals. There's been a lot of work, early slime molds, there was a lot of work on what causes these plasmodium to migrate. And, that's been studied chemically, and that's has a pretty interesting history. You can get certain slime molds, some of the cellular ones like dictyostelium you take a pipette and you have them in culture, you can drop out a micropipette, cyclic AMP, and it will migrate to it, along against the concentration gradient. It's one of the first kind of chemical signal things. That work was done back in the 60s. So they've been a model organism for a lot of basic biology, apart from all the taxonomy that we're talking about here. So a very interesting group of organisms. So Mike, how about the second one, so I don't take up too much time here? There we have it. I also saw this thing referred to. So this is Chromelosporiopsis coerulescens. I think like the pigment in an artist's oil box called cerulean blue. It had this beautiful sky blue color, which is what caught my eye. And it was on leaf litter in mostly a hardwood forest in a state park. Well, that's a third one. Yeah, yeah, that was a scene years ago. I didn't really mean to send that. I think everybody's seen that. That's that thing that grows on. This is on a hickory nut. I'm forgetting the name of it. Hymenoscyphes or something.
Yeah, Hymenoscyphus.
But this, I thought this was interesting. It got me trying to look up this blue pigment in fungi is interesting, and apparently there are many different molecular pathways. It's not a single pathway. The psilocybin mushrooms will stain blue if they're injured, and that whole pathway has been worked out in detail chemically, and that has something to do with um, that reaction of very similar to, it uses phosphatases to make an indigo-like pigment in psilocybin, but in this group, it's unknown. Some blue pigments in fungi are actually related to the synthesis of melanin and some of the precursors of melanin, which goes through tyrosine originally. And then there's also polyketides that are involved in other blue pigments. I couldn't really understand if anyone had any definitive evidence as to what the significance of these blue pigments in fungi are, whether it's a warning sign to predators, it's an advertisement. This particular organism apparently often has white, and I think you can see that here. It's a member of the Pseudomerulius. It's an anamorph. The telomorph is not known, so it's the asexual stage.
In the middle there,
What's that?
right in the middle, Doug?
Yes, right in the middle. You can see this grouping of these little star-shaped or sunburst-shaped. This is very typical morphology for this, from what I could see on the internet. But apparently, with age, it'll develop a pinkish and can even turn brown later on. So early on, it's blue and white when it forms the anamorph stage that we're seeing here. I just thought the pigment was beautiful. It's something, everybody's seen these sky blue pigments in fungi. I know there's an ASCO with it. I forget what it's called. I've seen them. They're little cups, cyano something or other.
Oh, yeah.
And it's just a very striking pigment. It would be nice if people were to study. I mean, there is a lot of, I went down a rabbit hole trying to figure out the pigment. And there's a lot of work on using fungal pigments to replace the denim industry, which is apparently very polluting. They don't use real indigo for dyeing jeans anymore. It's synthetic, but the chemicals used are extremely polluting, and it's $1 billion industry. So the DOE has given a grant to major universities to look for substitutes for indigo dye, and some of them have used bacterial and fungi species and try to bioengineer them to produce less toxic ways of producing blue jeans, which is interesting. I suspect that fungi are going to be used much more in the future, not just as we've seen for building cheap structures, that are sturdy, as everyone has read about, but also for the dye industry, which is apparently quite a large but polluting industry. So it's kind of an interesting sidebar, just because I like the color. I don't know much else about this organism. If I suspect other people have seen it, maybe someone could chime in about it.
Is it something that might grow on scat?
I don't think so. I think it was just growing on the forest litter.
Yeah, hardwood debris is what this thing likes. I'm pretty sure.
Okay. It's so beautiful.
Yeah, and it's quite striking when you're just kind of walking around and all of a sudden, whoa, there's a bunch of blue stuff on the ground.
Exactly. You're not sure what it is at first. Your eyes attracted to it.
Yeah,
It's small. I neglected
it is
to say it. It's
go
very,
on.
very small. Each one of those is, I don't know. Well, you see that the elliptical leaf with the elongated tip, they're probably only, half a centimeter at most.
Yeah, I was actually just about to mention. I was under the first time I found this thing. I thought it was a slime mold. It's very slime mold-esque. It wipes away quite easily.
Yes, yes. That's what I thought when I first looked at it. But then I started looking more closely and researching it. Exactly.
Cool.
And
Anyway,
as often happens when I'm examining a photo, even of my own, like something like this, what's going on in the bottom right there? You see that kind of more violet color, maybe kind of more flat or disc shaped.
yeah, I see that. Yeah.
Yeah. There's some more of it even to the right and up just a touch.
I I
That's
mean, it's
cool.
hard to say. It's probably the same organism, and it's just, something to do with the production of the pigment, and it could be you know, what the chemical sequence in. I don't want to speculate. A lot of dyes, of course, if you change the pH or will change color very, very significantly. You go from yellow to blue with a small bump in pH in a lot of dyes if you've ever worked with dyeing fabric. So maybe in the way the color is made by as a secondary metabolite of this organism. I'm not sure. I don't think it's a separate organism. Do you?
No idea.
Yeah.
I'm 50-50 yes or no, to be honest with you.
Yeah.
Could
Yeah.
absolutely be part of the same thing. Different stage that I've never kind of seen before, but it could certainly be something else.
Yep. Yep. It's hard to say. That's a good pickup. I didn't really see that. Good eye.
And do you have any experience with using fungi as a dyer for wool or fabric?
I mean, I've seen demonstrations. I haven't done it a lot myself. I've dyed. I've used indigo dye from the plant, Indigofera, and you use that with a dyes. The modern day dyes are usually have a reactive chemical group, and they're fibroreactive. They covalently bond to the fabric, whereas indigo, you have to use a mordant at a very high pH, like pH 10, and it just forms it forms.
That's how it works with fungi as well. If I'm not speaking out of line, right, you need a mordant with fungi dyeing, Lila.
Yeah, most of the time a mordant will help to get a better color and a more lasting color, I think. There are a few that will give you some color without a mordant, but most of the time the mordants are used. Alum, tin,
Yes.
copper less so because of the toxicity. Iron is used.
Yep.
Yeah.
A lot of reducing agents, but
Let me give a plug for the dye workshop, by the way, since we're on it. Sorry to interrupt.
Yeah, that's where I was going. I was hoping you were going to pop in and plug that. I was trying to set you up.
Yeah, yeah.
Oh, well, when is that? I was unaware of that.
Yeah, May 10th.
But
And
where is it?
Flemington, at a member's home in Flemington, New Jersey, and it sells out within an hour. So it'll be announced at roughly 8 p.m. on Wednesday. What is that? April 16th, I think.
That sounds interesting.
Yeah.
Is that selling out mostly to people who dye wool rather than to mycology interested people?
Well, yeah, there's a group of people that they're into knitting and crocheting, and they like the idea of doing it with fungi dyed wool. So yeah, but they're members of the club. It's only for members, so different people are welcome as long as you're a member. So stay tuned for that.
What's interesting is a lot of these natural dyes, not just from fungi, of course, the color you might see in the living organism is not what you'll get once you extract it and use it to dye and then use a mordant. It often changes color radically.
That's true, yeah. And then changing the pH, you can go from one color to a dramatically different
Correct.
color with the pH change.
Yeah, which is fun. I'm not sure how fiber fast many of these are. Mordant dyes are really forming a precipitate between the cellulose fibers. So there's usually some kind of, compared to covalent fiber reactive dyes.
Yeah, I don't know anything about that chemistry, but what I've heard, and I'm really a novice at this, but what some of the more expert dyers have said is that the mushroom dyes are actually more color fast than a lot of the vegetable dyes
Yes, that's interesting. Yeah,
and plant dyes. So
Yeah.
yeah,
Yeah.
it's quite an interesting area.
it is interesting. Well, I'll have an eye out. Where is Flemington? Where is that? Is that far from Philadelphia?
It's north of Philadelphia.
I'll find it. Okay.
Yeah.
Thank you for telling me about that.
Sure. It's where the Lindbergh baby was abducted.
I see. That's its claim to fame. Okay. All right. Okay. Thank you, Mike, for allowing me to present. Thank you.
Thank you, Doug.
Yeah, you're welcome.
So Tristan, would you like to share your screen?
Sure, I could do that.
It's always great to have somebody share for the first time. That's exciting. Yay!
Is that working?
Yes?
Okay. So my name's Tristan, longtime viewer, first time sharer. I went on a couple forays last season, and then I really picked a horrible time to get into this because it was right around the time that the drought started, and then it went right into winter. But I was excited to find this at the end of December, on a tree in Bordentown. And obviously I'm new to this, so if I use any terminology that isn't right, if somebody could correct me, that'd be great. I mean, it looks like I guess they separated out Enoki and this one. I know they used to be called the same thing. They used to be thought to be the same thing, but I think they separated them. So now I guess this one is Flammulina velutipes. And that was my best guess, but the only thing that threw me off was on the margin of the cap here, all the, I guess they're called striations. All the other pictures I've seen don't have that. But other than that, it looked pretty close. And then the other thing was that the gill attachment looked a little weird to me based on all the other pictures that I saw. It looks like it's not even really connected, but I'm not sure if based on the other pictures that I saw, it didn't seem quite like this. That one, here's a bunch growing on the tree. I know even less about trees than I do about mushrooms, so I have no idea what kind of tree this is. There's the gill attachment again. It looks a little strange.
You got great photos of it, Tristan.
Thanks.
Yeah.
There's
Useful photos.
Yeah.
Yeah, I've been.
Oh, you're downtown. Look at that.
Yeah, it's right around where I live downtown, and I've been doing a lot of like minor studying of how to take photos and watching videos about that kind of thing. So, in the past, my photos might not have been so good, but I think I'm doing a little bit better with that. So this is the tree.
Yeah.
I don't know if this helps. That's part of the tree. Like I said, I don't really know anything about trees. And then took a spore print, and it was white, which obviously matches.
Oh, good.
Matches Flammulina velutipes, but those other two features kind of made me a little bit unsure, even though it's probably right.
You got a nice heavy spore print there.
Yeah.
Which is that's a very Flammulina thing. I agree with your ID. I think your ID is good here.
Me too. Happy to see the spore print though. That's reassuring.
That's a classic growth form of it too, coming out of the tree and the bark like that as well.
Right.
Yeah.
Yeah, I don't know what the tree is from those buds or bark, but
Is there a better way that I can figure out what kind of tree it was? I mean, other than the bark and like if there's no flowers on it or anything, right? I don't know.
yeah.
To go back to your earlier point, that it is a useful thing to document. When you were showing it, you said, "I don't know if that's useful or not," but yeah, I mean, right? It is useful to some degree.
Yeah, for somebody who knows trees well, buds help.
Okay.
Yep.
Buds and bark.
Yeah, that's all. That's all that was really there for the tree. So that's all. That's all I could do.
Right.
Um, you're in Philadelphia, is that what I saw?
Bordentown.
Bordentown, okay.
Yeah.
I'm attending because I'm terrible at trees as well. Everyone in this group who's here often knows I do not know my trees. I am going to a winter tree ID workshop walk on Saturday in Philly with the Philadelphia Mycological Association. So,
Okay.
if anyone's interested, you have to sign up for it, but it's free.
When is it?
This Saturday in Philly.
And the people leading it are very good. The guy's arborist by profession, but very good at helping beginners get a good start on figuring out how to ID trees.
Well, that's all I have.
Man, that is some first share.
Yeah, it's a good first share, and also I get why the striations, which you are correct. That's what those are. It is a little weird, but not every mushroom is going to match the definition right in the book. There are environmental factors that could be coming into play, like this thing might have dried out at some point and then rehydrated or something like that. But yeah.
All right.
Yeah, it looks a little dry to me. So that's what I would attribute that to. And it tends to dry from the edges of the cap in. So.
Okay.
Yeah, while we're here, I've been reading Michael Quo's description, and he does mention that the cap does have its margin becoming lined. So when they get older, that's not unusual. So
Right.
Great share, Tristan.
Thanks.
Gold star.
Yeah, good stuff. Thanks for sharing.
Yep.
Okay, Christopher, you're up next. Are you ready? Are you there, Christopher?
Oh, yeah.
Okay, cool. Ah, we see your screen. Oh,
Weekend we were out there. Is that way better?
yes.
that's great.
There you go.
Beautiful.
And then we were by that Brendan T. Bryan forest.
Yeah, Pine Barren's favorite right here.
Great photos, Christopher. The texture of the caps on this are so beautiful, and the color.
Wow.
Look
What
at
did smell? So smell report before.
that.
Like none, and that goes for me not trusting that sense all that much, honestly.
Yeah. Okay. But that doesn't. So this, everyone, all the descriptions say this thing stinks, right? I've maybe only found it three. I see it all the time, and maybe only 2 or 3 times has that smell ever been present. Out of the maybe 15 or 20 times that I found it and smelled it, I'll see it from afar and like, yeah, I know what that is. But yeah, it's not as reported as every description will tell you this thing stinks.
Yeah, I honestly, because then it was, sitting on the table for a while, just nothing.
Except those couple of times that I did find it when it stank. Oh boy. Oh man. Like literally like walking up on a rotting corpse in the woods. If anyone ever has that smell in their brain, that's what they smell like.
Delightful.
Gross.
I wanted to say it had this like perfect white bit all the way around. I guess like as it dries out the hairs and the how enrolled the margin is. It had an all right amount of spores coming off. They have that slight tint of color to them, but almost white.
Yeah, but no hint of orange, right?
I mean, like there's a hue of some sort of pinkish,
pinkish, yeah.
whatever. This was from I took some off the Phyllotopsis here.
Oh, the hairs.
Yeah.
Yeah.
And it said that there might be some clamps. I think I got one in this one.
Yeah, looks
Yeah,
like
there it is.
there it is.
Yeah.
Yep.
Nice.
And they're just kind of, the smaller you go, just kind of squiggly out there. And there was a bunch of like this is on the gill edge. A lot of them were like kind of flat at the tops.
Interesting.
Yeah, I don't know. Maybe.
What was your process in mounting this? Did you do a smash mount?
Yeah.
Did you use a razor blade to chop any of that material up?
Yeah, a little bit, but then I kind of just kind of moved it around to loosen it up a bunch. Because it didn't want to really like smash out nice.
Yeah, I've seen a similar thing that confused me for a while. That I've always just at this point I chalk up to that's where I severed that with a razor blade.
Yeah, I don't know. There was too many of them like that to like.
Yeah.
But I got at least one Bisidia that came out of the moving it all around.
Yep. Yep.
The little allantoid spores. These things are pretty small, honestly. I thought I was getting like a 3x7.
Is this at 400?
Yeah.
Yeah. Pull it.
Which,
2 microns per deviation. Between 2 and 3 deviations. Yeah.
Yeah.
I'd love a review on how to do the measurements on the spores somehow.
Well, I mean, yeah. There is a bit that goes into it. The first thing you need to know is the magnification of your lenses. So if you have a 10x and then a 40x and then a 100x, you can kind of do the math, but like I have a 10x, a 43x, and then 100x. So when I'm doing my measurements at 400, I'm actually doing them at 430 magnification. So that kind of throws all the math off, but that's the first step, is knowing the magnification of your lenses.
And so, like, if you knew the magnification and you have the ruler like Chris does, like I have, then I mean that is as I mean the simplest way, right?
Theoretically, right? But then every machine is different, right?
Mhm.
So,
I have
so the
I
way
have,
your eyepiece kind of magnifies the reticle can play into it. There's a bunch of factors. So
So you have to do the
you have to calibrate your microscope, right? You have to
right.
calibrate on your machine how much each one of those integers, how many microns each one of those integers is at each level of magnification.
And you do that once or
Yeah,
not?
yeah. I mean,
Just one time. Yeah.
once you do it once, unless you're like me and you do it once and then figure out you did it, you find this knob that never turned, and then you get your microscope cleaned and find out that's a zoom knob. Then you have to do it again.
Yeah, okay. And so, but once you do it, you're supposed to remember something, right? Or no?
Yeah, what a lot of people do is they write those numbers down for the different magnifications and tape it to the side of the microscope, and then you've got it there.
Okay, and so once you've done it, and if you did not write those down, you should just like do it again, basically, and write them down, or
Right.
okay, yeah, I really need help in that area, big time. All right.
Do you have a stage micrometer? I think we've talked about this before.
Yeah, I think so. That's the ruler,
the one that's on the
slide.
slide.
Yeah.
That's the reticule in your field of vision here, but yeah, the slide, special
Yeah, I
slide
do
is the
have
stage
one of those.
micrometer. Yeah. Good.
Yes.
Good.
But I've actually done nothing with that though. So
Okay.
yeah, so I don't know what to do with that really. I need help. I need to get it figured out. Big time. So anyone wants to tutor me, I'll pay you.
I need to get my hands on another micrometer. Igor, maybe you want to mail one to me.
You could borrow mine. You let me know.
All right.
I should just give you mine because it's just sitting.
Because one thing I didn't do the last time I wrote down all my measurements was I didn't take a photograph, because that's what I should have done. I should have taken a photograph like I normally do when I'm taking my microscopy pictures of the actual reticle. Then I could, forever and always just go back to that. If you lose them, the measurements on the side of the microscope.
I did that. That's what I'm looking for in my photos right now. I took one at 100. I on the 10 on the. Oh wait, is that it right there? Oh yeah. You know what? Let me start to share again. I'll show you real quick.
Yeah, cool.
Great.
Yeah, it's a down night. We can get into these type of discussions.
I will say, Hunter, I've spent some time looking at some YouTube tutorials on how to do this stuff, and there have been some really helpful ones out there.
Oh, Emily, if you ever find them in your in your saved watch list or whatever, send me some, because I have watched a lot of them, but some are so much better than others.
I agree.
Yeah.
I'll see if I can find them.
Yeah, especially on this subject, that would be really great. Thanks. I did get sort of not a true dissecting microscope, but it's like one of these new fandangled, they have this like big screen that it's for like welding, I think, but I think it's going to help me out to do my sections and stuff like that. So I'm excited, and maybe just see things, see things that are tiny. That's like an in-between. I'm excited about it.
Sounds cool.
So my reticle is a 10x, and this is with the lowest one, the 10 times.
100 power here.
Yeah.
Okay, my reticle's a 10x too. I do know that.
So you can see how different. I laid it almost perfectly on. I got it like centered on there. And you can see how tiny. It's a little bit out of focus on this one, but how many little bars. So I took these to calibrate it. And then this one I think was at 400. I got to go back and do it over again. But this is what you want to do.
This is all coming back to me. Thank you for this.
Yeah, and you can see yours lines up pretty nicely.
Yeah. Yeah, but that's basically it. You're going to try to line them both up together so you can get that differential, whatever it is.
Yeah, so this is at
That's
100.
1,000. I guess that's 1,000, right?
I honestly, I didn't,
Yeah, because it's
Correct.
important.
Yeah,
it's important to
'cause
realize that each division
div-
in the reticle in the stage micrometer is 10 micron because there are 100 divisions.
There you go.
So this the big lines is the stage micrometer. So that's 10 microns, and you have your reticle lined up between those divisions, which means that each single tick in that reticle is 1 micron.
Yeah. Yeah, and at 1,000, yours lines up pretty spot
Perfectly.
Yeah.
Yeah.
But it's important to basically move your field division across the entire scale and see if that
Keeps doing.
measurement is consistent across, because it's going to drift and you won't actually maybe get even the average. So I do it very carefully. I basically explore the entire scale and calculate the value of each division, and then zoom in and do it regionally and see if it matches and maybe average it out. So I have a reticle that has 200 divisions, so I can measure 2.5 micron, but the actual value for mine is 0.465, turned out to be. Not 0.5.
Yeah, and that's important when you're really getting into the weeds, right? Of probably for you, Igor, Amanita spores, and trying to figure out if those spikes are half a micron to a full micron, things like that.
Yeah, for Russos, which I haven't done in a long while, because pretty much Russula microscopy. I mean, we don't even know what these things are with the DNA, so forget about the spikes. Yeah, but if you have large spores, it's less maybe relevant. If you have smaller spores, then you have to be more careful, especially if they are ellipsoid, which can be distorted because depending on how the spore is oriented in a field of vision. Is it tilted? Because if it's tilted, then you're not seeing things properly. That's why it's important to choose a specific view for the spore, which preferably is the side view as opposed to view from the up or down. Okay, that's too much information. But yeah, when you calibrate
good.
your microscope, make sure you record the values on a piece of paper, and so you don't have to do this again unless you're changing your lenses. So if your lens becomes cloudy, you have to get yourself another lens. Then you have to redo it for that lens, because the lens could be different, could have different properties, and the value of
As a beginner, I mean, when measuring spores, I mean, which objective should I be using?
I mean, ideally, you want to do fine measurements at 1,000 in oil.
Okay, ideally, yeah.
But ideally, but you can probably get away with some stuff at 400.
Okay, and so Chris was just showing spores at 400, was it? Or,
Yeah, 400.
yeah, they seem big. Um, really big though.
Yeah. So in that regard, at 400, each division is going to be 2.5 microns, ideally, but it could be different.
Okay.
Yeah.
Thanks. It's time for me to get back to it. Thank you.
Thanks for showing that stage micrometer photo, Chris.
When I did it, I was like, I'm not going to remember any of this. I was right, by the way.
Yeah, post-it note on the side of the. I look at that post-it note every time I'm doing the math because I would forget the number 2.23. It's a random number, but that's how many. I have 2.23 microns per integer at 400. Well, 430.
And you could see in that photo too, like how wandering around trying to focus down the reticle I was, which I'm working on getting that tightened up, a new jig for my phone. Because as soon as you start wandering, that also messes up. Like you could be not focusing properly because of that.
So, thank you very much, Christopher. Brendan, you're up next. I did put in the chat a link to the tree walk in Philadelphia for those who are interested.
Nice. Yeah. Thank you for that, Mike. You got
Sure.
to it faster than I could. So I just let that lie. All right. So I haven't been out in the woods very much recently. It's just been too windy. Like I'm scared to be in the woods when it's that windy. But I found this today. We were in the 70s today. It's been very warm. And I showed something similar last week that I was calling, oh gosh, Lachnellula. But I found it, or something in that realm again today, on a standing dead pine tree at the edge of a bog. So these in situ photos are pretty bad because it's very small, and I'll show you what the host was. Yeah, so it was on this tree, on this bark, and it was on the backside. So if you can see how small of an area, I was just trying not to fall in the water, pretty much. But I was able to get some collected, and that's what they look like under the stereoscope. And they're very much these, there's a couple other things I talked about last week, but I'm pretty sure this is Lachnellula resinae, which grows on pine resin, pine sap. And these things were between 1/4 millimeter and 3/4 millimeters, these little discs. But this was cool to find it on standing bark. Because normally when I find these little discos, they're on a fallen log, normally the underside of a log, but yeah, that was kind of cool.
That is super cool.
And then I'll share this, which I've shared before, things in this. I just call them a family at this point. Like, I'm pretty sure this particular fungus would fall into Jackrogersella, but I still don't think these things are the literature, the understanding of these things. They don't match a lot of the things that I see in the field. So this again is a little tiny ascomycete. Let's go to an in situ photo to give you a little bit of an idea. I mean, each one of these little kind of dome-shaped clusters is half a centimeter max, maybe 3-4 millimeters to half a centimeter, something like that. But they do grow into each other. One of the ways that I tell these things apart is whether there's the presence of an astiliar disc, which I'm learning is more and more subjective the way that you look at it. I would say these things do not have a disc around each one of these osteoles. And each one of these little holes is kind of where the spores come out. But like, look at the one in the center there. Maybe there is a little bit of a disc. So, yeah, even this one here.
Brandon, did you break it open?
I did. I didn't take a photo of it. What were you looking for? The shape of the parathecia?
No, I was wondering if it was like pink dust inside.
Oh, no. So inside is a series of kind of cavernous, at this point, parathecia. So the spores are getting shot out of each one of these little individual holes at the top, and they were all empty. So when I say cavernous, I just mean pretty much empty little cavities, more or less.
And it's black inside.
White inside.
White inside.
Yeah. Well, a layer of black, then a layer of white, and then a layer of black. But I would call the flesh of the thing white. But I think you guys have heard me talk about these enough in the past, and they are somewhat confusing. And I'll do the microscopy on that and maybe update us next week.
But I also was growing on. Sorry, did you say what that was growing on?
I didn't say what that was growing on, and I don't know what it was growing on. I would say it's definitely hardwood. I think I took a photo.
Sort of beachy.
Beachy, but also birch is not out of the question, maybe.
Okay.
But I said that with a question.
Yeah, I don't think it's a birch. It could be one of those which one is it? The hornbeam or the hop hornbeam? The one that's also known as musclewood.
Right.
Okay.
Right. Yeah, maybe that. It's very smooth.
Would you lean beech here? Because I didn't see a lot of beech in the area, but there were a few. It is smooth, so
yeah. The thinness of the bark kind of pulls me away from beech a little bit. It seems a little thin, the bark.
Yes.
Okay.
Yeah, I'm not sure if I'd hazard a guess, but it might be one of those two that just mentioned.
Yeah, one of the hornbeam things. Yeah.
Is that like how big is that piece of wood?
1.5 inches, 2 inches.
Okay. Yeah, because a lot of stuff when it's that young is smooth at first.
That's true too. Yeah.
Right.
Some maples are smooth like that. Yeah.
Mm-hmm.
I was curious, like, what is it related to? Like, it's an ascomycete, but where would it fall? Like, with other?
At one point, all of these things probably would have been hypoxylon.
Oh, okay. Makes sense.
And it, I don't know the exact taxonomy. It's probably still in Hypoxylanaceae. Jackrogersella, Annulohypoxylon, a couple of these other things. I think they're all in, the Hypoxylon family. But yeah, Annulohypoxylon, Jackrogersella, it's one of those two genera, and they are recently split from Hypoxylon. So, yeah.
Okay, thanks.
And I wanted to show that, which I'm pretty sure is Jackrogersella, to show this, because when I found it, I was under the impression, this is much smaller than the last thing that we looked at. This is probably 3 millimeters max wide. But you can see it still has osteoles, parathecia kind of shooting through that upper crust of this ascomycete. But I don't think this is Jackrogersella or Annulohypoxylon. And I don't think I have the technical, I don't have the vocabulary to kind of explain why. Other than each one of those osteoles on the last thing that we looked at, the little beak looking thing, the kind of pinpoint coming out the top, there was only one of those per kind of more bulbous stromata. And these stromata, I thought, were discus, kind of looked like a mass of discs together. But once I got them under the stereoscope, you can see that's not the case. It's just a bunch of these parathecia kind of shooting through an irregular fruiting body, I guess is the best way to put that. And then this is the host, which that's probably maple, right? We can look at the leaves on the ground. Yeah, not knowing my trees, maple was my first guess, or oak.
Yeah, oak maybe. Yeah, some of those other leaves down there kind of don't look like oak or maple.
I think that's a smilax leaf.
Okay. That I don't know anything about.
I learned it recently.
Did you try the app yet?
Say it again.
That app for the trees?
I did. Didn't work all that great.
No, you didn't have. We get a pretty good success rate with that thing.
I might not be looking at the right thing. I don't know, but
my wife just snaps a photo of the bark from like 2 feet away, and it does this thing like, oh, I'm scanning, and then spits out an answer, which it's not always right, but not
good success rate. All right, and then
Smilax is a vine. Okay.
Yeah, it's sticker bush.
Yeah, like a greenbrier. Okay, I didn't know that. All right, cool.
Let's go back to that. Would you agree that could be this leaf here?
Maybe. With my 10 seconds of knowledge I have here.
Yeah.
Remember smilax usually smooth?
Yeah, I think most of them are smooth, but there's a lot of species of it apparently. So maybe there's some that have that more serrated edge. I don't know.
Yeah, we talked about this briefly a couple weeks ago on some kind of rust I found on a leaf.
Yeah.
And it was all about like the ribbing, I believe, coming from the base of the leaf, and multiple ribs.
Right.
But
Yeah.
I was looking for things to share as Mike was asking, if anyone has anything, and there weren't a lot of answers kind of starting out. So I went back to previous observations from March in previous years. Did I share this at the best of this year? Does anyone remember if I shared this
so.
You think so?
No.
If not, no, I don't think so.
All right. So let's have a look at it because it's really cool, I think. So this is Pseudomerulius aureus. This is a thing that grows on pine logs, normally on the underside. That's where I found it. You can see these kind of toothy, irregular. They're more folds than they are teeth or pores. Like it's just kind of folded up, and they make these kind of structures. But it's got a beautiful.
Wow.
Oh yeah,
Wow. Yeah.
yeah. It's a burn associate as well. So you can see that this is on a burned conifer log, the underside of a burned conifer log. And it's got that very Merulius thing where it kind of radiates out, right? But yeah, the colors on it they were gorgeous, bright yellow, deep kind of burnt orange to red.
Beautiful. Look at that.
Yeah, isn't that neat?
Oh,
Wow.
Why is it that everything that grows on pine has got these orange tones?
Yeah, that is pretty interesting, right? I will say, I'm sure, Chris, you were probably thinking of Phyllotopsis nidulans when you said that, right? Which you shared earlier. I just remembered, and I wanted to mention it when you shared that. That thing grows on oak as well. It is not strictly a conifer associate. Phyllotopsis nidulans grows on hardwood also.
It's not like a chicken that it's like a very different one. It's the nidulans grows on both.
Mm-hmm. Yep.
Is there another? I'm sorry. Is there another Pseudomerulius that's on hardwood?
Yes.
Is that the Curticii?
Curticii, right? Yep. And that thing can be more lobate, kind of forming more shelves, kind of cap and shell thing. Which you can see this has actually, I've seen observations of this that does the same thing. And you can see that this is a little bit reflexed from the surface where that cap would be. So all this was the direct bottom of the log, and this is where it was slightly raised, so it gave itself a little bit of space to reflex away from the wood and make that kind of shelf shape, more or less. But yeah, I think Curticii is a little more apt to make shelved fruiting bodies, if I'm not mistaken.
As far as I know, Pseudomerulius curticii grows on hemlock and pine, not hardwoods.
That could be. To be fair, the only knowledge of any Pseudomerulius, I only found this name while researching this, and I found it only once ever. So that could very much be the case, Igor. Maybe that's also a hemlock conifer thing.
Yeah, I'm not saying that yours is curticii. I'm just saying that for curticii, the host is a conifer, not a hardwood.
Okay.
Oh, do I have that backwards? Has anyone looked up aureus? What's the host for aureus? Maybe I just found a name for this thing and applied it.
I've never found areas before. I've never heard of it either.
Barkless conifer wood, especially pine.
I want to find that.
Yeah,
So
and
pretty.
Jan's a pretty good identifier. She agreed with it. I think is she a member of our club? Jan?
I don't think so.
All right. That'll do it for me. Is there another thing I wanted to share? Nah, that's cool. I'll let somebody else go. Thanks, guys.
Okay, we're up to you, Hunter. Just a minute here. Let me get set up.
Okay. Yeah, I just pulled some stuff out of my mushrooms to ID folder. And I feel like I might have shown this before, but when I first started out, I wasn't set up to take good notes when I got IDs from people, and sadly, I may have to repeat some, but I'm clearly, we don't have a lot of information here, but the color staining is so pretty, and I'm wondering, is that like a late reaction? Like, I mean, I didn't cut this mushroom. I found it like this. I came across it like this, and so it could have been. It might have seemed blue at first and then turned this purple. But well, one, maybe it is a common color. And I mean, have people seen a reaction like this color in a bully? It's clearly a bully, even though we can't see the pores, but.
And those colors look like something that some like AI came up with.
But it's definitely not. I promise you that. I found this, and so this was on the bluff of Lake Michigan, probably middle of summer, a year or two ago. And they get some good stuff there. It's a little bit more like Wisconsin, what they might get up there than the rest of Illinois, but I just thought the color was stunning, and I thought maybe, oh, it would be a clear ID based on that, but maybe not.
I'm not convinced it's a bolete.
Okay, yeah.
Yeah, it's kind of looks like gills there on the left, doesn't it?
Well, look at the one on the right is more telling. You see one flat gill running the whole web.
Yeah.
Okay.
I think that's a gill mushroom.
I was starting to question that too as soon as I saw it up here, blown up. But I just sort of assumed it was a bolete because of the reaction and the kind of the layers. It just looked like I guess gill layers look similar to tube layers in a lateral cut like that. I mean, not similar, but there's the other main flesh of the mushroom above it that I guess can look kind of similar.
Yeah, I mean, I wouldn't be. I've been shocked by tricholomas before in my experience. Like when you flip, oh, that's a Pseudomerulius, and then you flip it over, like, oh no, it's got gills. So maybe something like that. But that's just a guess.
Maybe one of those russulas with the reddish bottoms.
The gills or the pores. are obviously purple, right? Is that the color that I'm reading? Am I reading that correctly?
Yeah, I would call that purple.
Well, I thought it was a stain reaction only.
It's interesting.
Yeah, I mean that was my impression, and why I took a photo of it in the first place, because you can kind of see stain going up into the
flesh, kind of like a graying purple.
Yeah. But I mean, I may be wrong. So if those were purple gills, I mean, could it be a Laccaria? I mean,
that's what I mean. That was what I was thinking. But the flesh is way too thick for a Laccaria, right?
Ochropurpurea.
Oh,
maybe
that's one I don't see that one often.
Okay, I mean, because that's a thick mushroom, the stem, the cap, but the cap color doesn't look like it at all. I mean, and that's what made me think bolete too. It just kind of had that sort of bolete color to me, the cap, but okay. So yeah, probably I should just delete this.
No, you keep it up, keep sharing it until someone says, I think I know what that is.
Oh, I don't know about that, but I'll keep it. Yeah, maybe it'll become apparent down the road. Yeah, right. Yeah, it's a terrible picture.
Igor is here. Igor, do you think this thing's a bolete, or do you agree it's potentially a gilled mushroom?
The context, right? That white context does kind of say bolete, but those purple areas on where you think the gills or the pores would be seem gill-like to me. I don't know. Igor, are you there?
What about Cordonarius? Like, I'm looking at the base of the stem right now, and
that's a great
another webby kind of thing or something down there too, right?
That black, it looks almost like it's
Cordonarius is a good idea here.
I don't know. You didn't save it, dry it by any chance, did you?
No, I mean, no, I should not even put you through these observations. I haven't been getting out, and there's really nothing yet out except like scarlet cups and a few little things, but nothing too exciting. So, and I haven't been out, so I like to try to get labels on these photos in my folders, because they're sort of meaningless otherwise. So the next ones at least are better photos. So we can move on, but if anything occurs to you,
Ochropurpurea is a pretty decent fit for that last one. I was just looking at some observations, and there are some brown capped things that are at least labeled as Ochropurpurea, but without some better photos, I think it's mostly speculation.
Yeah, it surprises me because that is a mushroom I know really well. So to be totally confused, like in the field, that it like to not pick up on that, but at the same time, the cap color is really variant on that mushroom. So maybe it's clearly not in great shape either. So, well, thank you. This one I think is a fairly recent photo, but from this last season. I don't really have any more info on it at the moment. But it's kind of cool, and thought someone might have some ideas on it.
That
looks like
one in the bottom right-hand corner. Is that part of the group here?
I think it probably is, but I can't say with any certainty.
Younger one, maybe.
Yeah.
This looks like some kind of a Gymnopus in the Droathilus group.
Yeah.
That would be my guess.
Yeah, I was just Googling sulphureus. Is that it? So
Yeah.
Okay.
what is the name of that thing? Is it Gymnopus sulphureus?
Is that the jack-o'-lantern mushroom?
No, not the jack-o'-lantern. That's Amphilutus.
Yeah,
Yeah,
that's
definitely not that. I know
I
that
think these caps, I think, have been, that they look waterlogged to me, so they're kind of discolored. I think
Yeah.
it's a lot of rain.
Gymnopus subsulphureus is a thing that doesn't look dissimilar from this.
Okay. Mm. Sounds good. I'll look that one up. Yeah. Sorry. And the gills are free, right? On that one? And this here?
Well, they could be, notched or,
Okay.
uh,
Kind of hard to tell.
yeah.
Michael Kuo says they're attached to the stem narrowly, very crowded, yellow to yellowish.
Okay.
Yeah, and to Igor's point, maybe you picked Igor. You threw Gymnopus out there. That stipe looks very Gymnopus to me.
Yeah, kind of a semi, kind of a translucent. Yeah, has this kind of a ivory sort of feel to it.
Okay, I'm looking at this now, and yeah, I think this is a good lead, and probably what it is. I'll try to pinpoint a little more details on where and when I took it, which I should be able to do, and then match it up. But I've written that down, so hopefully I'll decide to label it that. So thanks on that. Can we go to the fourth one?
Yeah, while you're going to that, just quick back to Bianca's thought, just to explore the jack-o'-lantern Amphilotus alludens. That would be a darker color, more orange, and the stipe would be fatter than appears in that picture, and the gills would be somewhat decurrent, running down. They wouldn't look like that. So, and they would be darker, but it would just generally be a darker mushroom.
And although these are clustered, I don't think they're cespitous. I don't think, but maybe, no, they're not.
Yeah, okay, that's another thing about the jack-o'-lantern. It is cespitous, so.
It looks like these maybe could be actually, but they're, it's not like, I think, a requirement. I don't know.
Yeah, we can't say, I don't think, from this photo, but.
No, not from this one, but I'm looking at other ones.
Okay.
But
of the things that I've called, right, of the Gymnopus subsulphurans that I know, they are always clustered but not connected. So what, gregarious, I guess.
Yeah, but not true cespitous. Yeah.
Correct.
Okay.
Yeah, there's little patches of them, but their own individual mushrooms that are connected in any way. So
Right. And these are probably terrestrial, or are they, do you think, growing on
litter, I think. I think most of these type of things are terrestrial for sure. But I think they're saprobes.
Yes.
And Omphilatus will grow, it is growing on wood.
Definitely, yeah.
Yeah, the jack-o'-lantern, right.
Can be buried though.
So, yeah,
yes.
Buried roots for sure. Yeah.
Often is, right? Yeah.
Yeah, probably most often when I found it. Yeah.
Yeah, a lot of the time. Me too. Okay. And so then this Lepiota, I think. I thought maybe someone had seen the Elsa talk, because I feel like she talked about this one in that the annulus is so distinct. This is an old picture, and so it's not great. And I don't have the proper shots, but the annulus seems to be almost like enough to put a name on it. But I'm gonna have to go back and watch. I feel like she may have talked about it. Did anyone else remember or know or have a different feeling that it's not, wouldn't have been included in her talk on parasols and lepiodas?
Zero opinion.
Yeah.
I don't know these things.
Okay.
Tell us why it's not an Amanita.
I think it's just the way the veil is, but that's a really good question. And without seeing the base, I think, you know, that's some missing information. But you know, a lot of the lepiodas are similar. I mean, they look similar. They have annuluses, and there's a lot of white ones. But I don't know. Love to hear someone else say. So maybe it is. Maybe
I'll
it
say
is.
that skirt is very skirt like.
Yeah, big time
Right?
skirt. Yeah.
Yes, it's so definitive. But I feel like I remember her citing this one species, and it had this skirt, but
I don't remember. I feel like I did hear her talk one time, but I don't recall.
okay. Well, it's available, and you guys can watch it.
Yeah.
And I will watch it again, and that sort of brings me to my next observation, which is in the
Also, just real quick on that last one.
yeah.
The gills are so tight. Look at it.
Mhm.
Yeah.
And look white to me, with
Yeah.
no discoloration.
Yeah.
So what's the idea here? I kind of stepped away.
Oh, well, we haven't come up with one. I was pushing for some sort of lepiota, based on the annulus, and this idea that I thought Elsa had cited this one in that latest talk she gave, but then I haven't gone back to verify that. And I had been looking at a lot of amanitas lately, so I mean it could be, but I was leaning toward lepiota and thought maybe the veil alone might scream a certain species, because it's a pretty cool veil. I mean, it's really
I think
neat.
this is Destroying Angel.
Okay. Sorry.
Maybe if this is from the south, maybe something elliptosperma.
Okay.
Pinkish gills.
Just another destroying angel to
Yep,
throw
just
into
another Destroying
my-
Angel.
Okay,
Look at that stipe, very textured. It's very, very common for these things to have such a textured stipe.
I was gonna bring that up. So that is a common feature with destroying angels. Interesting. Okay. All
Oh,
right.
and I just noticed. Can you zoom in on the base? It looks like you might have some material there.
Yeah, there's a tiny bit of maybe. Yeah, it does. And to the right too, like it's, yeah, you're right.
Yeah,
Kind
that's
of coming
that's
out.
the edge of the vulva.
Right.
That's the one of the limbs, I guess. Yeah.
Okay, just another one. Okay. Thank you. And so I have this one more, and it's a Gyromitra. It was growing on the wood, and I feel like I might have shown this before, but it was growing right on the wood. And I am relabeling all my Gyromitra now based on the Dirk's talk. And I don't know if people saw that, but the names have all changed. The species names are pretty much the same, but now the genus has been broken up into a lot more. But I still don't know which one this is. I did rewatch that, and I could have sworn he mentioned one that does sometimes grow on wood, but then when I kind of rewatched it, I didn't hear him say that about any. But I've never seen one growing really right out of wood like this. I mean, I guess I have. I have another one like that, to be honest, but I don't know which one does this. And I was curious. I mean, I know I'm very into themes and other people aren't, but maybe we could do like a morel. And I know you guys do morel. So usually we could do morel and false morels and just maybe, or at any rate, I plan to share more pictures of them because I want to try to get the new names down. And I think there's a lot more out there that haven't been named yet, and I'm excited to try to find them.
I was looking in after watching that talk. I watched it too. I was looking around in my my new Latin dictionary, and I looked up mitra. It says a headdress, a turban.
Cool.
That
And
makes
then
sense. That makes
gyros,
sense.
like spinning, right? Like
Yeah, like a
Well, like the only route I could find to that had something to do with like a thing that you would run a horse around, or like almost like more like a verb, like going around. So like a turban that goes around.
oh, cool. Yeah.
Mitra, is that something that an archbishop wears?
No,
Yes.
Well, miter,
that's a miter.
miter.
probably derives from the same,
Mit-
headdress.
Yeah, right.
Yes.
We talk about some spores being mitriformed, right? Mitriform. So
Yeah.
all a common root.
And that's literally, I think, named for the hat.
Yeah.
Yes, a miter's hat.
Yes.
The spores, I loved how he presented each genus, with each species, and then had the spores right on the, like he'd have a photo like this and then a little box with the spores. And I took screenshots of every single one, and it was very inspiring in terms of wanting to get that microscopy going and try to match them up and see. And so I'm hopeful.
You got to ask Dave about these things.
Yeah,
If
I
you
know.
noticed, he got a shout out during the presentation there.
Yes, he had found two of the discoid ones in the same location. And there were like no names on them until then or something. Yeah, pretty cool. Yeah.
Yeah, and to get to the base of this observation, I don't know what this is. I didn't see that presentation. I've never seen something that that looks like a Gyromitra to me, but I've never seen one on a tree. So
Yeah, and so it wasn't standing. I will say that it was laying down. It might look like it's standing, but it's not. It's
Okay.
laying down. But
it
still,
definitely looks like it's standing. So thank you.
okay, yeah, because the way it's vertical, not horizontal. Yeah, I should have spun that around. But I actually have another one growing out of wood. I'll show it next time. It's very degraded though. But it's a different, it might be a different species. It appears to be. It's really like stipate, I guess, and the other one, whereas this one didn't seem to be, but I guess that can vary with those a lot. So I'm liking to focus on them personally, and I'm going to be a little bit. So that's all I have, and
to the those in charge of TT, are we planning on doing our kind of annual Morel review, more or less?
Yes, I think we'll do it in April.
Yeah,
Cool.
probably early to mid-April, somewhere in there.
Sounds good.
Yeah, speaking from my own experience, and it's a singular experience. I don't know what kind of morels other people find in New Jersey, but I find that they are less abundant than other parts of the United States. I don't think we get as much variety, but I could be wrong on that. We don't have a lot of elm. We don't have a lot of. I don't go to the apple orchards to look for morels. Maybe they are abundant there, but they're not suitable for consumption, so I don't even look. But
Does the club ever go to the Princeton Waterworks anymore?
No.
There used to be a ton of morels there. People would come out with baskets.
Yeah. Yeah.
Used to be. That's the operative word.
Yeah.
I mean, those woods have been decimated, and after all the ash trees died, good luck in the competition. I think the last time I went there was 2015.
Uh-huh.
Yeah, we stopped having forays because it was just a madhouse. We're gonna get so many people.
Yeah.
And of course, all the locals will probably scoop up all the morels before us. I mean,
Right.
they would know when they come up based on experience. So yeah, we decided to eliminate that foray from our schedule permanently.
Somewhere out east, they now have found a one species of burn morels, right? I've heard that. And they might not be out now, I mean, in the same time, but probably.
I don't know, but we're yet to sequence a strange morel by Emily Rollins.
The one by the fire.
Yeah. I
Yeah.
remember that observation. Yeah.
Me too. Yeah, so that hasn't been sequenced yet. That'll be interesting.
Yeah, because I mean, I track burn fungi and conifer, in the pine barrens. I've never found a morel in the pine barrens that wasn't associated with like a rogue apple tree. And even at that point, it's like one.
Yeah, I was just going to say, like, I've been putting together this idea that I've been everywhere that I have found them. We got to look at the geology map, because if you draw a line from like south of Princeton going more towards the direction of like, if you drew a line from like Princeton to Bordentown, and then like that followed that arc kind of up, you find them on the western side of that line in a way different abundance than you do on the other side.
So that's the coastal plains, right?
Yeah.
It's the inner outer coastal plains in South Jersey. I find them in the outer coastal plains. That's where I find them, and they're associated with tulip trees, Liriodendron, tulipifera, and no other tree consistently in my experience. And again, it's a limited experience. I'm absolutely not in this for the fungi for food game. But every spring I do like to have a morel meal, and that's the only place that I can find them is with on the outer coastal plain with tulip trees.
Tulip trees are great for morels. I love hunting in those. There's not enough of them. Never enough. I know soil can be important, and it sounds like maybe there's like kind of a drip line that maybe in those coastal areas too.
I'm sorry, a what?
A drip line, like
Oh, from like off the side of the tree?
like the coast, the fog or something. They have
Oh,
it on the west coast. It's like a drip line that goes to the mountains, I guess.
Yeah. Well, I don't. Again, I'm a South Jersey guy. North Jersey is a different world. They have Piedmont, right? So it's a little more mountainous. They have different tree associations and things like that. But I don't think we have a drip line down here in South Jersey, at least as far as I'm aware of. And I do know what you mean. It's like a microclimate coming in from the coast, right?
Yes. Right. Exactly.
I don't think we have that, but I
Okay.
could be wrong.
Yeah, maybe because the direction of the weather going west to east primarily would maybe go against that, maybe, probably. Okay.
Well, I also think the West Coast. I don't know do they have an outer coastal plain, or does it go mostly, especially like in Northern California and things, northern, northwestern U.S. states, Oregon, and things like that. It's more
Totally different. isn't it?
mountain dominated, kind of
Yeah.
right on the coast. I
Yes.
think.
Yes. Yeah, and it's like waves, like right on the coast, like right, not these like intercoastal plains like you're talking about, or
Yeah, I think those coastal shelves are more actually underwater out west. You know what I mean?
mhm.
But again, I'm kind of talking out my rear here.
Yeah, no, I know what you mean, like, but you guys have a lot more of maybe, what are they called? Like the coast of North Carolina, Outer Banks and stuff like that. Do you have that or no?
similar.
Similar, yeah. Which they don't have it all out west, like, I don't think, yeah.
Well, the thing that we have in South Jersey, it's such a unique ecological community, because it is high. The soil is so highly acidic. I mean, it's sand. It's sandy soil. 20 miles east, 20 miles west of the actual beach. You know what I mean? That it's all that acidic pine dominated ecology, which is a pretty unique
Nice.
ecological community in whole, right? Doesn't exist many other places.
Sounds very much so.
So I'm going to interrupt here. I think Lila has an observation or a few to share. So I want to give her a chance to
Good.
share.
Okay. By the way, I think coastal plain is kind of an Atlantic coast and Gulf of Mexico thing in North America. I don't think there's coastal plain on the West Coast, but that's a better
Agreed.
question for Jason.
Yeah, Jason was a better answer to that question.
Yeah. All right. What do I have here? Now, let's see if anybody knows what this is. Found it about a week ago. The color being more yellow is what is bugging me, I think, about these.
That's a good picture.
Thank you.
Irpiciporus.
Yeah, I was thinking it's some sort of Irpiciporus. Is it mollusc? Is it? I don't
Yeah,
think. I don't think it's pachyodon, but
I
the.
think that's the same thing.
Oh, are they the same thing? Mollusc and pachyodon?
I think
Like,
so.
it's like supposed to be white tooth, but these are quite yellow. Maybe it's just because they've been out in the winter and that's just how they're aging. I don't know.
It's got a certain toasted marshmallow to it.
Yeah, it almost looks like the I don't know if you call that a subiculum, the white, or just a mycelial mat, I
Yeah.
guess is a better way to put that. But yeah, you can see, it wants to be marshmallowy.
It's got like stiff peaks, like some meringue. I'm sorry, I'm fooding this up, but it just that's what it's doing for me.
Okay, yeah. All right.
Could you put that name
A
in the
sweet
chat
tooth,
for
huh?
us, Brandon?
Sure. Which one? Irpiciporus?
Yeah.
All right.
And what was that growing on, Lila?
Dead tree, I think. Let me see if I look at it. I actually think it was at the base of a dead ash, but I'm not positive at this point.
Uh-huh.
Could have been an oak, sort of looks oaky. Was dead.
I feel like I've seen that a lot and should know what it is. And
Yeah.
yeah,
I don't know what this is down here. This is something else now I see in the corner, right? Or is that the tree? That might be the wood itself down there. Okay. All right. So, all right. Irpiciporus was what it was.
Pachyodon mollis. Well, to be fair, I don't really know what's going on with the taxonomy there.
Yeah,
But
one
I
of
think
those.
yeah, I've seen what I call or used to call Irpiciporus pachyodon. I am now using mollis, but that's just me.
Okay.
I've seen it with yellow hymenophore before.
All right.
Especially when young, and
When young, really?
and I don't think I've ever seen it in this state where that subiculum is so fluffy looking. But normally when they are mature, there is a layer of subiculum between these, shelves shooting out.
These
So
toothy areas. Okay. All right. All right. Good.
yeah,
Thank you.
that right there, that's classic, right there. I think.
Okay. All right. Thank you. What else? All right. Let's see if this is marginata. They're kind of old. Super decayed wood. This stiping rather wide though seems somewhat inappropriate. It does look like there's a remnant of a ring there, and a younger one.
Just initially, this looks maybe an inch across this cap, maybe multiple inches.
Yeah, this one was bigger. I would say maybe an inch and a half. It's pretty good sized and just seemed rather robust. So I'm like,
yeah.
I've never seen marginata with such a thick stem.
Right. That's the weird
agree.
thing about it. So I don't really think it's this, but I don't know what else it is.
You got a gill shot of the cap?
Just these. Let's zoom in. Kind of old. I think it's bleached out. Obviously, very old.
Yeah, even the gills on the ones on the right there that you first,
this group.
yeah, those gills are very long.
Yeah, yeah. So doesn't really fit Marginata, but
So what
Like
else
an
could it
old
be?
Gymnopilus or something.
Yep. Yeah. Gymnopilus is a good guess. That annulus, maybe we can go back. Is there any evidence of color of the spores or anything?
I don't really see any color, and I didn't bother to bring this back and try to get a spore print. I probably should have. It's so old, I'm not sure there were any left.
What's the gill attachment on most Gymnopilus like?
Actually, I see some forking in the gills now. That might be a clue.
I think Gymnopilus is a good guess, Chris.
I'm really bad at those. All right. All right. I will poke around in Gymnopilus and see if I can find something. Thank you. Yeah, I had some other things, but I knew what they were. They were pretty boring. Nothing interesting.
Oh, I want to see.
You want to see more?
Yes.
Yeah, this is all good common stuff. All right, let me share again. It's just fun to actually get out and see something. All right, so here's our friend, the ceramic parchment, Xylobolus frustulatus, always fun to find.
I love it. I love that mushroom. So pretty.
And is this at the cut portion of a log that might have been on the side of a trail or something like that?
Yeah, along a trail here, you can see the end of the log. I think it was probably an oak.
It's crazy how much this thing loves that.
Yeah, it really likes that position on the log, doesn't it?
Yeah. Not exclusively, but yeah.
Yeah. Yeah. Well, and it probably, maybe we like to take pictures of it there because it's nice and flat and you really see the effect there. Maybe that's part of it.
Right.
Yeah.
But you can see that cool pocket rot, right? They were just showing off the upper right.
Yeah, maybe. I gotta
Oh, right.
Yeah. Up there, yeah.
Now a little to the left of that, where you were
over here.
No, the radial. See that radial, those straight lines.
Yeah.
So see how there's a little bit of pocket going between those lines.
Oh, you mean some depth in there? Yeah.
Mm-hmm. Yep.
Yeah. Okay.
This thing does it causes a really interesting rot, which I've been paying attention to lately.
All right.
Cool.
All right. Yeah.
Is it a pretty effective decomposer? Is it pretty vigorous? Do you know?
Yes, very much.
Really interesting.
Yeah. And it renders so that pocket rot. What's really interesting about that is a lot of other brown rots or white rots, different fungi break down their material in different ways, right? This one, it does not. So it breaks down either the lignin or the other one.
Cellulose.
Yes, one of the two. The one that it doesn't break down, it doesn't really even affect. So the wood is still super hard. When you're trying to dig into this, it's like chipping away to rock sometimes when you're trying to find the pocket rot. So when I find it like this, where that pocket rot is exposed, I always think that's pretty cool, because it's tough to actually get down into the thing to find it, because it's so hard.
Interesting.
Cool. All right. Something soft and fleshy. We had some rain. Look at this.
Beautiful.
Fresh.
Yay. It's coming. The mushrooms are coming.
They're coming, yeah, out of the dead. This is oak. I always find this on oak twigs, sticks, small branches on the ground.
I love when the light shines through that. It's so pretty.
Yeah.
I've been finding a lot of like liquid ambars, like younger ones that are just like coated top to bottom in it.
Yeah. Cool. So is there anything else? No, the lichen, Hydnoporia. This is everywhere too.
Oh, I'm happy to see that again. And yeah, that's good.
Kind of out of focus picture. What's this one? That's a little better. Yeah, it's just all these things out there that you didn't know about, and then all of a sudden you start to notice them and you see them everywhere. It's kind of fun.
Yeah, it really is. Yeah, that one I just learned this winter from you guys.
Okay. That's it, I think. Those are the things I've seen in the last week. Oh, another Fuscoporia gilva, but I've shown that before. Versicolor. Yeah, that's it.
No, what's the thing you had labeled as fungi?
Was there another thing I didn't? Okay, let me look. Yeah, it was that Irpiciporus thing.
Oh,
Picoporus thing, yeah.
Yeah.
All right, and this is not Galerina. I gotta look for Gymnopilus, something like that. Trying to get some
Good
lichens.
luck. Good luck past that.
Yeah. Oh, here. Maybe you know what this one is. This is a total guess. I thought it was a diatribe kind of
You're
a
not
thing.
You're not sharing your screen.
Oh, sorry. I got off of that. Let's see. Try again.
There we go.
Yeah.
Wow, that would be a pretty big fruiting of stigma.
Yeah, this stuff is tiny. You can't even begin to see this with the naked eye, these little osteoles or whatever they are. Yeah, these are big patches, like
feet.
Yeah,
right?
like almost a foot. This biggest one is probably a foot long. This one's 10 inches or so. And there's little patches, but I don't know. I thought for some reason, what did I go to?
Diatribe stigma would be the first.
Yeah, I looked at that, but then I looked at this thing, Camilla punctulata.
Nice.
But I don't know if that's it or not.
I looked at that recently. Yeah, it's more patchy, right?
Yeah.
Look at that. That's a neat pattern there.
Yeah, but that little picture down the bottom, of that one. There was like that little pincushion look.
That looks
Yeah.
pretty good. Camilla.
You know, this it really looked like this stuff. to
Yeah, yeah.
Yeah, right there.
me. Yeah.
Cool.
And this one, yeah. So I don't
Yes.
know.
Yeah, that looks good.
Yeah. So that's what I put on it. So I guess I'm going to stick with that for now.
And what was it on? Did you
Hickory.
mention
It's on
hickory?
on
Oh,
hickory.
cool.
Yeah. Yeah. I probably shagbark, but it could have been a different type of hickory as well. I'm definitely on hickory. So
That's a neat one.
yeah, so that was different. But all
And
those
right
colors.
and right on the heart, not quite heartwood, but you know, shy of the bark.
Yeah, it's not on the bark, it's on wood underneath. Like, yeah.
Cool.
Yeah, here you can see
Do
some
you
of
think it
these.
broke down the park first?
Well, yeah, that's the question. Was it inside and it came out and it made the bark fall off, or did the bark fall off? I don't know.
Yeah, you could. So on the one on the left, if you lift that bark up,
This piece here.
yeah, it answers that question.
Yeah, see if it's under there or not. Yeah.
Mm-hmm.
Yeah. All right, well,
cool find.
Yeah.
thanks. Next time I'm in that area, I'll take a look at it again and tear that log apart a bit more and see what I find.
Pretty cool. I'll have to start looking for that.
All right.
Yeah. Yeah, I think I've seen it before and I just wasn't even sure it was fungus, it's some of these things.
Oh,
same.
so many of these diatribe type things, I mean, especially if you're in a burn, a recent burn, that's what they look like, right? It looks like some caught
Yeah, it looks
fire
like
for a
charred
second.
wood. Yeah.
Yeah.
Right. Yeah, exactly.
But there's so many of them. Once you start paying attention to them and the different hosts and, where to look on the actual either trunk of the tree or canopy branches on recently felled things. I mean, it's such an extremely diverse world of fungi in different families that aren't even related to each other but look exactly the same. So it's pretty cool, and that's a cool find.
Yeah, I'm glad you shared that.
Good.
Thank you, Lila.
Thanks, everybody.
Yeah. A good evening. Some interesting shares. So I wish you all a good night, and we'll see you next week. Same time, same place.
You too, Mike. Thanks for hosting.
Sure.
Yeah. Thanks,
Thank
Mike.
you.
Bye, everybody.
Mike.
Night all.
Good night.